or the Q&A pod. Cliff and I will answer them at the end of the session. Tomorrow, you'll be mailed a link to a short, voluntary, confidential survey that we hope you will fill out because it helps us bring these webinars to you. The email will also contain information on how to obtain CEUs for viewing the live webinar. This webinar is being recorded and will be available on the EABU page of the emeraldashborer.info webpage. Thank you for attending. And now we're going to go ahead and get started. And before we begin, I did want to mention there was a little bit of a mix-up with the host plant identification part. I do have some host plant ID information for you, but we're going to be sharing some recordings that Carrie Tauscher did a few years ago. So there's going to be a little bit of a switch in the middle, but we'll still have all of the information you need about thousand cankers disease. So with that, I'm going to go ahead and get it handed over to Cliff.
In this presentation, I hope to give you an update about the latest news about what we know about thousand cankers disease. So I thought it'd be a good way to start by hitting you with a punchline. Thousand cankers disease is really not an imminent threat in this part of the country. Despite the presence of the insect vector and the pathogen in this region and in Indiana, the disease just simply is not yet expressed. Extended periods of drought, however, can promote the expression of the disease, which is the rapid death and decline of black walnuts. So what we'd like you to get out of this presentation is they want you to be able to watch for and report rapidly declining stands of black walnuts, especially during a drought. I'd like to start by acknowledging the contribution of my colleague, Dr. Matt Ginzel, who's a forest entomologist, who has an appointment in the Department of Entomology and the Department of Forestry and Natural Resources, and currently serves as the director of Purdue's Hardwood Tree Improvement and Regeneration Center. So let's get back to thousand cankers disease. One of the signature expressions of this disease is the rapid decline of walnut. And when I mean rapid, this is the time frame that I'm talking about. On this photo on the left, you see a green black walnut with just a few yellow leaves just in the upper middle of the canopy in June of 2008. In September, in the fall of that year, we noticed that there are some branches that are already lacking leaves, and it's starting to leaves are starting to yellow. And by June, the tree on the right, which is the dead black walnut, has completely died. So in the span of a year, it goes from green to dead. That's what we call rapid decline. So what causes thousand cankers disease? It is caused by a beetle vector, and that means it's a beetle that moves a fungal spore called from a fungus called Geosmithia morbida. And when it brings that spore into a susceptible host, a black walnut, and the environmental conditions are correct, the disease flourishes and the plant starts to die. The beetle is required for disease development simply because the spores themselves have no other way to get from tree to tree unless the beetles are carrying the fungus to the bark and boring inside, okay? They readily taste the branches, and this tasting activity is what spreads the fungus. And you can see that the cankers on the upper right of the slide here, these darkened areas are rather small, and they're confined around each individual penetration of of the beetle. So it's not the beetle that kills the trees. It's actually the fungus that grows in advance of the beetle feeding that clogs up the vascular system that causes the tree to die. So the beetles, as they feed, the fungus will sporulate and making little seeds in the tunnels, which are picked up by the beetles. And when they move from tree to tree, that's how it moves around. And it's the fungus which actually kills the tree, not the. So, it's called thousand cankers diseases because there are literally thousands of beetles, each causing small cankers on under the trunk, and that's what actually kills the tree. So if you look at the penny on the right, you see that each walnut twig beetle is about the size of Abraham Lincoln's ear on a penny. And in these two 18-inch logs, they actually collected 23,000 beetles, okay, in 1 year after they were cut. So the reproductive capacity of these beetles is quite impressive. So each colonizing twig beetle will release a pheromone that will summon other beetles to attack. But what happens is that you can see each brown spot is at the center. There's a little hole where the beetle has been feeding. So each beetle causes a small canker. And when you have thousands of beetles, you have thousands of cankers. And it's these thousands of cankers, like the one you see on the right, where they all coalesce, that destroys the vascular system. And that's what kills the tree. And so you see the tree on the right that's just been peeled, has been killed by the 1,000 of little cuts. So 1,000 cankers, it's almost saying that the tree is being killed. It's as a death of 1,000 cuts. So one of the questions that came out early on was, can other beetles besides besides the walnut twig beetle pass on 1,000 cankers? There are quite a number of other beetles that are actually found on black walnuts. So there was a study that Matt Ginzel and his colleagues were working on where they harvested 4 black walnut trees that were symptomatic, that had symptoms, that had the rapid dieback. They took sections from different parts of the tree and they reared them inside buckets. And they collected and counted the number of beetles. And they found lots of beetles besides the walnut twig beetle. They found, several xylosandras. Just to say, there's a number of beetles here that are some ambrosia beetles. And there are a couple of weevils that are there as well. So this was kind of scary. So they conducted a much larger study in Minnesota, Illinois, Indiana, and Ohio, and North Carolina. And the Ohio site with the red dot, that is where they were working, where the beetle was found, and they're working in trees that had 1,000 cankers diseases. And in the other sites, they were looking at in forests where the beetle might have been present or the fungus might have been present, but the disease wasn't really expressed. And what they found through this study, where they repeated this over and over again, was that they were able to find Geosmithia morbida, the fungus, on quite a wide variety of bark and ambrosia beetles, a lot more than that I showed you in the previous slides. But very few of them had actually live propagules. In other words, the spores that were associated with those beetles that they picked up were not simply able to grow and reproduce on new trees. So many of these detections of Geosmithia morbida were found in areas where none of the trees were expressing the symptoms of the disease. They weren't expressing that rapid dieback. So it's quite likely that the Geosmithia morbida fungus is widely distributed. And it's also possible that many beetles will move it around. But just because the beetles are associated with a fungus does not necessarily mean that they're going to be spreading the disease. So in order to get to the bottom of this question, Matt and his colleagues set up a project where they were monitoring the health of thousand cankers disease symptomatic trees and surrounding trees in Butler, Ohio. And they looked at 40 trees, and they wanted to compare how the decline that they were observing was manifesting itself in comparison to how it occurred in the West. And as you can see in the slide here, the photo on the left, you see the thinning black walnut trees, and you see it's a different pattern of thinning than the one in Maryville, Tennessee, that also had thousand cankers in the east. The one in Colorado, the entire canopy is quite thin, whereas the Maryville, Tennessee, the canopy thinning is mostly on the top. So it suggests something somewhat of a difference. So when they started looking at at the amount of crown that was left over, how green the tree was left on a year-by-year basis, they noticed with the top line, the dashed lines and the squares, that those trees that did not have thousand cankers disease did not decline much at all. As a matter of fact, their quality improved from 2015 to 2016. They were a little bit greener, and there was no substantial decline from 2016 to 2018. In 2015 to 2016, there was some decline, but it wasn't really significant statistically, and it pretty much remained the same. So that even though there was some slight decline in the amount of live crown in the trees that were symptomatic in 2015, it wasn't a great rapid decline. So the conclusion of this study, from these 40 trees that they looked at in the Butler, Ohio site, was that there was really no spread of decline to the new trees in eastern study sites. Only the thousand canker disease symptomatic trees declined. The disease was localized only to those individual trees. The rate of crown decline in these trees at the epicenter of the infestation as a whole did not differ from that in other study sites in Butler County. So basically, there's no widespread decline of black walnut trees, at least in Butler County, Ohio, where thousand cankers disease. So another hint at this they thought they'd look at was try to compare levels of precipitation. Because we know the West is much drier than the East, and they wanted to compare the level of precipitations with the times when they saw new thousand cankers disease progression and development, new rapid death of black walnuts. So in 2012, when they looked at in Richmond, Virginia, they found when precipitation was low, there was a lot new spread and progression of thousand cankers disease. But in 2013, when they had a lot of rain, no new symptoms of thousand cankers were observed. And another study site in Knoxville, Tennessee, where there was high levels of annual precipitation, there was simply no new symptoms. So they really think that precipitation really has a lot to do with it. And just an example of what was going on, take a look at this top slide of a photo in 2010. This tree had 60% of the crown was left alive, and there was some moderate dieback of branches associated with thousand cankers disease. But by August of 2013, that tree had healed itself. So clearly, there was really no progression of the expression of the symptom, despite the fact that the beetle, the walnut twig beetle, and the fungus. So what we're finding is that ecological factors, including environmental conditions, are likely to play a role in the development of thousand cankers disease through the expression of the rapid death, and climate differences between the western states where thousand cankers disease is actually being expressed, and on eastern black walnut, could explain why we don't see it here in the eastern states. So in summary, many beetles and weevil species were found with Geosmithia morbida on their bodies. The fungus is there, but few had live propagules that could actually be transmitted. There was actually some detections of the fungus in states where there was absolutely no known instances of trees dying rapidly from thousand cankers disease. Thousand cankers disease symptoms are likely to occur when there are periods of below average precipitation, and trees will recover like they did in that Knoxville, Tennessee site as the precipitation increases. A thousand cankers disease will not progress in the absence of the primary vector, the walnut twig beetle. And finally, you're going, if even though we do not see much in the spread of thousand cankers disease, we still want to keep our eye on it just in case something odd happens. So if you see rapidly declining walnut trees, especially during a drought, this is something that we would like you to report because this might alert us to some changes in the situation.
All right, and now we are going to switch over to the host plant section of the talk. And again, I appreciate all of your patience since we did have a little bit of a mix-up. But I will get that open right now. You'll still get the same information about the host plants.
Yes. So walnut is another tree that can be easily confused with ash with its big compound leaf. For identification purposes, there are some really easy things that you can look at when trying to identify walnut and separate them. The first is that on this large compound leaf, there are more leaflets, and the leaflets vary in size. So they're generally smaller at the tip of the leaflet and smaller at the base of the leaflet, so they're smaller at the apices. You also get this really interesting situation where there are two small leaflets at the tip of a walnut leaf versus ash, which generally has one. And the other really easy identifying characteristic that can help you is that the buds or the leaf or twig arrangement on walnut is alternate. So the buds kind of zigzag down the twig rather than being right across from each other. The buds on walnut are naked, so they have kind of a white fuzzy appearance, and they tend to be kind of right in the middle of the forehead of a monkey or ET, depending on when you grew up. So down here is this great leaf scar. It's got two little eyes and a happy little smile, and the bud originates right in the middle of its forehead. So in leaf off season, you can identify walnut from the buds. It can be similar to some of the hickories, but because of the jugglins in walnut, these little vascular bundle scars are going to be kind of a dark brown color. So that's something that you can use for helping for identification in leaf off. The bark pattern on walnut, depending on the age, can look very similar to ash. And I generally don't recommend scraping or picking on the bark of any tree. But in this case, taking a knife and gently scraping or just gently picking with a fingernail, when you fleck off that outer layer of phloem, you can see that the bark or the phloem cells that accumulate creating the bark are a dark chocolate brown, whereas the ash in the photos from Cliff with the woodpecker flecking was kind of a buffy beige color. Your walnut is going to be dark chocolate brown. So that can be a helpful identifying feature. Just another bark image here, a little more blocky than ash. The flowers will be coming soon, and they also are kind of a helpful identifying characteristic for walnut. The other thing is if you can find a twig on the ground or remove a twig from your specimen, if you can very carefully with a knife or a bypass pruners clip that twig in half, the pith of each twig on a walnut is going to be chambered. I tell people are, what is chambered pith? Chambered pith looks like the underside of a mushroom cap. It looks like the gills. And that's very unique to walnut. So between the naked kind of grayish fuzzy buds, the ET faces on the twig, the large compound leaves, and the chambered pith, you should be on a good course to identify walnut in your backyard. If not, these guys should help you out, right? So the fruit of walnut is relatively large. I would say small baseball size. They have a smooth, kind of pimpled textured skin on the outside of the husk. And if you rub on it, you get that quintessential kind of citrusy walnut smell. So please do, if you have a labeled specimen, make sure you get very familiar with that fragrance. It will help you with identification later. The only other host that I want to bring up is butternut. And this is pretty rare. Not everybody will have this in their area, but if it is, they look kind of just like a weird black walnut. The chambered pith exists, but it's much smaller. The leaves are slightly cupped on the edges, and the leaf surface, the upper surface of the leaf on butternut is a little shinier. Generally fewer leaflets, but it will still have a similar fragrance, just a smooth bark on a young butternut. The twigs, again, look very similar. The ET faces are a little bit smaller, but with that little bud right at the top of his forehead. And the fruits are really, for me, the characteristic that helps give it away, other than the shiny leaf surface, is that the fruits on butternut are smaller. They're kind of like a tiny lemon. They're very fuzzy and sticky. So on butternut, you're going to be basically looking for similar identification characteristics as walnut, but with slightly smaller size and a little bit different texture. So hopefully that is helpful to you all, and I'll send it over to Elizabeth.
All right, and now I'm going to wrap up with some reporting information. Again, as with these other talks, I know that those of you that have been listening into all the other talks in this series have heard this a lot by now, but we keep repeating it because it's so important. If you do think that you have seen signs of thousand cankers disease or any other invasive species, please do report that. There are a wide range of ways you can report. The first is with the website eddmaps.org or edmaps.org and its associated set of apps. In the area that I'm in here in Indiana, we use the Great Lakes EDN app or the Glidden app. You really have to search for it exactly the way I'm showing it on the screen now, because otherwise you end up with a banking app and they can't exactly help you with invasive species. There are other apps for different parts of the United States as well as Canada. And there is also a general EdMaps app. The benefit of using the general app is if you're someone who travels a lot, it has all of the invasive species that are either here or of concern across the country in it. The benefit of using one of the regional ones is that it has just the organisms that are a potential issue in the area that you're in, or are a potential future issue in the area that you're in. And so that way you don't have to scroll through a million different invasive species. For example, when I look at the Great Lakes EDN app, it doesn't have things that are issues in Florida that wouldn't be an issue in Indiana, and vice versa. You can also call the number 1-866-663-9684, which is a phone number where you can report invasive species. There are also usually email addresses for different areas. For example, here you would email the Department of Natural Resources if you encountered a invasive species of concern that hasn't been found here yet. So any of these options are great ways to report. We also have a summary of how you should report invasive species on our website for Indiana, reportinvasive.com. This website also has resources and other information about invasive species that can be helpful. And then finally, I always say, if you forget everything that I've shared today, you can always just reach out to someone who works on invasive species, and there's a good chance they'll be able to connect you with the right person to send in that report to. It might, take a few emails in an email chain, but usually we're all talking to one another, so we know the right person to send you to. When you do make a report, there are a few things that are really helpful for us to have. First, please do write down where you are. This helps us go back and check the area later if we think what you found is a potential invasive species, or if it's something where we can't quite tell. The next thing is take a picture. Photographs are incredibly helpful for us for IDing invasive species. It lets us kind of get that initial assessment of the situation. And then finally, if you can collect a sample, that's really helpful as well. With cankers disease, that might be a little bit harder, but if you can get a sample, that's really helpful for us. And with that, I'm going to wrap up. Just as a reminder, again, we do have more information about all the invasive species in this series, as well as many more, including, plants and vertebrates and other things on our Report Invasive website. We also have a social media presence on Twitter, Facebook, and Instagram. Our handle is Report Invasive on all three of those. We put updates about invasive species on there. So if you're interested in that, please do give us a follow. If you want to contact any of us, our contact information is on the screen. We are always happy to answer questions. And now I will, let's see if Cliff is on. All right. If you have any questions, feel free to put that in the chat. I am going to start out with this one right here. Where does G. morbida exist in the landscape when not impacting black walnuts or carried by insect vectors, e.g. soil, plants, other plants, etc.? And Cliff, if you're talking, you're muted at the moment.
Okay, can you hear me now?
Yep, there we go.
Okay, great. Yeah, so G. morbida is an obligate pathogen of black walnut. So that means it would have to be inside the black walnut tree itself.
All right, great, thank you. Our next question is, how fast does it move? I understand that it was found around Philadelphia. I have about 90 acres of mainly walnut in North Pennsylvania near the New York state line, Bradford County. I'm concerned about it reaching my property.
Okay, so let's, I'll take that question in two parts. The first question is, how fast can it move? Well, this disease is vectored by bark beetles, okay? And so the bark beetle, as far as the bark beetles will fly, that'll be as far as it can move naturally. But if you are moving firewood around, what'll happen is that you would probably, it can move as far as the log is transported. So it's yet another reason why you should not move. You should buy your firewood locally and burn it completely. And when firewood is an important vector of exotic invasives. All right. Now the question is, what is the risk, I think, to your black walnut in Philadelphia? Well, based on the presentation that we put together today and the work that's been done on this, you remember that in order to cause a disease, you need to have the pathogen, you need to have the host, and you need to have the right environmental conditions. All right. Now, we have the pathogen in Eastern North America. We have got the vectors in Eastern North America, and we have the host. But we do not have the environmental conditions. So cankers disease is a very weak pathogen, and it requires a stressed tree for the beetles to successfully attack and spread the spread the cankers. Remember, it's called thousand cankers disease because the tree is killed by 1,000 by lots of small cankers, and eventually girdles the tree and causes the limbs to die. So because you're living in Eastern North America and it's not nearly as drought prone as the Western states in Colorado, where it has been, and also in the upper, in the Northwest, on the east side of the mountain ranges, where it tends to be much drier in Eastern Washington state. So you do not have the environmental conditions for 1,000 cankers to become a problem. So what that means is that the take-home point of my presentation was that you should only be you should be very concerned about 1,000 cankers when we have extended periods of drought. And with climate change, the summers are getting hotter and drier. The springs are getting wetter. So if you have an extensive drought in your area, that would be when I would look for it. So I would say that even though the pathogen and the beetles may indeed be near where you live in Philadelphia, I do not think that the risk is very high at this point.
Okay, thank you, Cliff. Let's see, more of a comment here. Given the progression of EAB, I doubt there will be many ash around to mistake for walnut. Oh, I wish there were more ash, but you're not wrong there.
Yeah, so so that was an interesting story. I don't know if Elizabeth, did you tell that story?
I did not. Do you want to share
Okay,
it?
yeah, sure, I will. So what happened was that when we first started looking for 1,000 cankers disease, somebody called up and they said, well, we got 1,000 cankers because our walnuts are dying. I said, well, I said, I don't want to be rude, but how do you know that they're walnuts? He said, well, we pick walnuts out of the black walnuts out of the field every year. I said, well, I guess I can't argue with that. So we had some Indiana Department of Natural Resource inspectors go to the site, and when they arrived, they started laughing because they noticed that it was a mixed planting of ash trees and black walnuts. So even though there were black walnuts on the ground, the dead trees were all ash trees. So that was you always have to be sure you're identifying your tree properly.
Yeah, it's an easy mistake to make with those big compound leaves like that. I don't see any more questions here. If so, I'm going to wrap up. If you have any final questions, feel free to still throw those in the chat. You can contact any of the speakers today to ask follow-up questions if you think of them later. We appreciate you coming to listen to our talk through Emerald Ash Borer University today. You will be receiving a follow-up email that will have the speaker's contact information, as well as information on how to get continuing education credits or certificates of attendance, if that's something that you're interested in. Thank you again, and thank you to our panelists. Everyone, have a great day. Bye.
Thank you. Bye-bye.